Everybody, in this video I am going to talk a little bit about the Drosophila P element. So Drosophila are fruit flies, and it's a model fruit fly often used in genetic research and molecular biology research. Now the Drosophila P element is going to be a class 2 transposable element. It moves by a cut and paste mechanism, and one of the reasons I want to talk about this element is it will come back later on in the course when we talk about an epigenetic mechanism involving these strange RNA molecules called pi RNAs. So the Drosophila P element is about 2.9 kilobases long, so 2,900 base pairs long, and it encodes a single gene, so a single transposase. Now it has, like other class 2 transposable elements, it has terminal inverted repeats, so TIRs on the ends, and they are 31 base pairs in the P element, and the Drosophila P elements are characterized by an 8 base pair target site duplication. And I'll go over the mechanism of how that target site duplication occurs, but it's very similar to what we already saw for the insertion sequences in prokaryotes. So right here I'm just showing that there is a transcriptional start site associated with transcription of the Drosophila P element, and there's going to be a termination site. Now the location of the stop codon is going to change a little bit, and I'm going to cover that when I talk about regulation of expression of this transposase associated with the P element. So I wanted to talk a little bit more about the terminal inverted repeats, because the transposase, I guess the P element is a little unusual in that the transposase, binds near the TIRs and actually makes one nick within the TIR and another one at the target site duplication TIR border. So here I have diagrammed the 31 base pair TIR, and I'm showing the transposase down here, and I'm curving it around because I want to fit all these base pairs in here. And obviously it's not to scale, right? This is 31 base pairs. This is going to be nearly 2,900 base pairs long, and then 31 base pairs over here at the end for the other TIR. I have the target site duplications, 8 base pairs right here, going to be shaded in green on the ends. Now, I guess the nick that is made within the TIR is 17 nucleotides from the target site duplication. So we can count that. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. Okay, 17 right here. So one nick is going to be made between these two A's right here, and the other nick is made between the target site duplication and the G, the first G right here of the TIR. Now this is an inverted repeat, right? So this TIR, let's say they say it's going from left to right, this one's going to be going from right to left, and we can see that CATGA right here. Down here we have CATGA. Okay, so just checking that, okay, yeah, these are inverted repeats. These are the same sequences, just inverted relative to one another at the ends of the P element. Now we can check that this cut right here, this nick, should be 17 base pairs from the 17 base pairs from the target site duplication. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. And then this nick right here is going to be right between the G of the TIR in the target site duplication. So essentially what we're getting here is a staggered double strand break, right? So these nicks are so close to one another that, okay, essentially we're getting a double strand break. They're just the nicks are relatively far apart than what we're used to seeing when we're talking about double strand breaks. So now the transposases are going to remove the P element. I have this written as transposase, right? I meant that encodes the transposase, but here's the P element. They're going to bring this to another target site in the Drosophila genome. And it's going to leave sequences like this at the excision site, right? So let's say this was somewhere on chromosome 1 in Drosophila. The transposases remove the P element and they leave behind these 3' overhangs right here. So this is a 3' end, 5' end here, 3' end here, 5' end here. And as we saw with the insertion sequences, this has got to be repaired by some DNA repair process, probably non-homologous end joining or that related microhomology mediated end joining process that we saw earlier in this course. So one of the neat things about, because of the way the P element is removed and carried to a new target site by the P element transposase, what one can do is one can look at the Drosophila genome, look for sequences that have the 8 base pair duplications, and in between the 8 base pair duplications, the remnants of the P element TIRs. So what is this diagram here? I forgot to mention. So what I'm doing for this diagram here is I'm taking these sort of 3' overhangs that were left after removal of the P element, and I'm just bringing them close to each other because we don't know exactly how non-homologous end joining is going to fix these, if that's the repair process that does it. So it could trim away some of these nucleotides on the ends. It can even add a few nucleotides between them. But essentially what's going to have to happen is that these are going to have to be brought together, and whatever bases are missing, we're going to get these gaps, and those gaps are going to need to be filled by a polymerase, and the nicks are going to need to be sealed by a DNA ligase. So I'm assuming here that there's no trimming, and non-homologous end joining is just going to link these two together and fill the ends. So all of this will be double-stranded. But back to the original thing I wanted to mention is that one can view or examine the Drosophila genome and look for these sequences that are characteristic of the P element TIRs, and if there are duplicated sequences. Now the target site duplication for the P elements, it's a characteristic, it's 8 base pairs long. So if we see 8 base pairs here, followed by some sequences that match the P element TIRs, followed by an identical 8 base pairs over here, well, we can be pretty confident that this is a P element excision site. So there used to be in that genome a P element at that location. So Drosophila genomes can be littered, if P elements were present in that lineage, can be littered with these P element excision sites, which is, I think, pretty interesting. So what am I doing with this thing here? Oh, okay, yes. So right here, what I want to use this to imagine that the P elements are, the transposases are taking that excised P element, which is this thing right here. Now we'll have transposases bound to the ends right here, and I could color those if we wanted to. How about I add just a little color? So we've got the transposases bound to the ends here, and they could be bound to each other also, just like we saw with the insertion sequences. It doesn't really matter. So they're going to identify a new target site, and at this point researchers think that target sites are chosen at random in the Drosophila genome by the P element transposases. So I just diagrammed what an 8 base pair sequence right here, A, C, G, T, C, C, A, T, and the ends represent any sequence to the right and left of this putative target site. Now, the transposases, when they choose a target site, they cut 8 base pairs apart. So we get another staggered double strand break. So those are going to be pulled apart like that. And the P element is going to be ligated in to that staggered double strand break site. So it's going to look something like that. So I'm kind of off screen, right? A little bit off screen, but you can see what I'm trying to point out, and you can see this should match the notes also. So we've got this big, so we have this A right here of the target site ligated to the G of the TIR, and this G of the this A of the target site ligated to this G of the TIR, and then we've got these gaps that need to be filled by polymerase, a DNA polymerase, and the nicks need to be sealed by DNA ligase. So I have a better diagram of that, I believe. Right here. Okay, so that same thing I just showed you with the stuff off the video, I just made a smaller diagram showing you the lower half of the target site here, the upper half of the target site right here. We have those gaps. We have the TIRs here. A of the target site ligated to the G of the TIR. A of the target site ligated to the G of the TIR. And then the 2900 base pair P element between these regions. So once this is filled, right, so this is going to be a 3' end, so this can be filled that way. This is the 3' end, so this can be filled that way by polymerase, and those last nicks can be sealed by a DNA ligase. Okay, so the P element has moved to a new location. And what did I not diagram here? I didn't diagram the transposases. And I'm assuming there are 2 transposases involved in this process, just like we saw with the insertion sequences. So, okay. So that's essentially how the P element moves, very similar to the insertion sequences in bacteria. Now I want to talk about P element regulation. So the P element is something interesting about it. It only moves in germline tissue. So the tissue that's in charge of making gametes, right? So all the other, if we're not talking, we can divide tissues into 2 types, right? Somatic tissue and germline tissue. So the germline tissue is in charge of making the gametes. Somatic tissue is essentially everything else. Now the P elements, the transposase to move the P elements, it's only expressed in germline tissue. And the regulatory process is pretty neat. So we go back to our original diagram of the P element. So the 2.9 kb P element, so it encodes a transposase. And this is where I said, okay, so there's a transcript from here. This is a transcription start site and a stop site. And if we take a closer look at that transcript, so after processing, well, let's say halfway through processing, we're going to talk more about RNA processing in upcoming lectures. But there's going to be a 5' cap. There's going to be a poly A tail. So we've got 2 processing steps done. But another aspect of messenger RNA processing or pre-messenger RNA processing is to join the exons together, remove the introns. So the P element, the gene, the transposase, is made up of 4 exons and 3 introns. However, this third intron is only removed in germline cells. So we have 2 different, I guess, transcripts, and this is like an example of alternate splicing. So in somatic cells express a protein that binds to the third intron here and keeps it from being removed. So the messenger RNAs that are translated, the P element messenger RNAs that are translated in somatic cells have all 4 exons, but they also have intron 3, right? It's kind of weird to think that a messenger RNA can still have an intron. I guess we can call this a retained intron. It wasn't removed. Because it's here, what happens when that ribosome starts translating at this start codon right here, and remember, so why is the start codon here? Well, messenger RNAs have a 5' untranslated region that precedes that start codon. So the ribosome starts translating, and then it gets down here, and there's this intron still here, so it translates and it hits a stop codon. So when a ribosome translates the P element messenger RNA in a somatic cell, because that intron is there, we get a 576 amino acid non-functional transposase. So it's too short. It's missing some key amino acids needed to function as a transposase. And there's some evidence to suggest that when this 576 amino acid protein is in the cell, it can prevent any full-length transposases, functional transposases, from functioning. That's why some researchers call this the 576 amino acid P element repressor. But that's kind of complicated, so we can just assume, we can just consider this to be a non-functional transposase. So that's why P elements do not move to new locations in somatic cells in Drosophila. However, in germline cells, so germline tissue, the tissue that's in charge of making sperm and eggs, gametes, so all 3 introns are removed, and we get a messenger RNA, a P element messenger RNA that looks like this. And then when this is translated in the germline tissue, we get the full 751 amino acid transposase. So when the full transposase is in the cell, in a germline cell, it can search for those P elements in the genome and move them to new locations. So I think that's pretty interesting, and I think researchers have identified this protein. I do not. I have intended for some time to look up the identity of this protein that binds to intron 3 in somatic tissues to figure out why isn't this protein expressed in germline tissues. I haven't had time to do that yet. So feel free to take a look at that if you're interested, and let me know what you find. So one more thing about P elements, and again, I alluded to why I wanted to cover the P elements in some more detail, because we're going to talk a little bit about a really important epigenetic mechanism towards the end of the course that involves piRNAs. So just a little introduction to that. So in Drosophila, there is, in other organisms too, but we're talking about P elements in Drosophila, so I'm going to explain this phenomenon called hybrid dysgenesis with respect to Drosophila, because we're talking about P elements. So in Drosophila, there is a phenomenon called hybrid dysgenesis, and dysgenesis, this term usually refers to abnormal development of the gonads. So tissues, those organs that, ovaries and testes that make sperm and eggs. So dysgenesis refers to the abnormal development of ovaries and testes and things like that. Now the hybrid part, so this is sort of like, if you remember from introductory genetics, how Mendel had true breeding lines of pea plants, tall pea plants. When they were crossed to other tall plants, they always made tall progeny. And dwarf pea plants, when they were crossed to other dwarf pea plants, they always made dwarf progeny. So the same thing happens with fruit flies, right? So scientists could isolate two strains. They had P strains and M strains. So these were essentially true breeding lines of Drosophila. And you cross a P to a P, and all the flies are P type. And you cross an M to an M, and all the offspring are M type. Now, there's something interesting that happens when you do the hybrid cross. So you cross P to M. And it only happens half of the time. So I'll explain that. So start with the easy ones. So if we take a male fly from the P strain, so a male P fly, and cross to a female P fly, so a male and a female from the P strain, the progeny are all, we're going to say, all the offspring are normal. Now the same thing happens with M flies. So you take an M, a male from the M strain, and a female from the M strain. I don't know why I put that backwards. I don't think that's a real symbol. Offspring are normal. Now, we take a male M, we do a hybrid cross here, we make hybrid offspring. The female is from the P strain. Offspring are normal. And surprisingly, however, if we take a male P fly and cross that to a female M fly, this is where we get the dysgenic offspring. Dysgenic offspring. So essentially, the gonads in these flies here, these offspring, do not develop normally. So, and after many years of research, scientists figured out that it's P element activation. P element activation occurs in the germline tissue of the dysgenic offspring from this cross. Now, one of the interesting things here was that, essentially the genotypes of these offspring over here and these offspring here are essentially identical, right? Because, we're just switching which of the parents, the paternal or maternal parent, was from the M-type or P-type fly lines. So the offspring, the genotypes are normal. But for some reason, the P elements are only activated and only cause a lot of trouble in these offspring over here. So I don't want to give too much away at this point, but essentially what we have is the P flies are the ones with the P elements. But okay, that doesn't really tell us too much, right? Because we have P elements right here in this mom right here, and we have P elements in this dad right here, but only these offspring over here show the dysgenic phenotype. And the idea behind, the explanation for the problem is that we've got, let's say, sperm from P flies And now the sperm, most of the sperm is made up of the nucleus. So there's not a whole lot of cytoplasm in there. Now let's say the sperm is from a pea fly. Now we have an egg from a pea fly. And this nucleus of the pea sperm has pea elements, because I already told you that the pea flies, those are the flies with pea elements in their genome. In the pea egg, here's the nucleus right here, so the blue represents the nuclei of the sperm and the egg here. Well, this also has pea elements. And let's look down here at the M egg. Now again, the M flies don't have pea elements, but they have nuclei, right? So here's the nucleus, no pea elements. So we have all this cytoplasm right here associated with the ovaries, well, the eggs, relative to the sperm, right? And the idea is the cytoplasm has an anti-P element factor. And and this one does not. So cytoplasm, no anti-P element factor. Now, why do the P eggs have the anti-P element factor, but not the M eggs? Well, because the nuclei of the P eggs have P elements. The nuclei of the M eggs do not have P elements, so there's no anti-P element factor in here. So when the sperm, which doesn't have that much cytoplasm here, and it doesn't have this, or if it has, it doesn't have sufficient enough of the anti-P element factor to suppress P element activation, when this sperm fertilizes, I was going to say infects, when the sperm fertilizes the M eggs, the P elements become activated. So obviously, I previously specified that the P elements are activated in germline tissue. So I'm guessing that as this fly starts to develop, it's the germline tissue that becomes mostly affected by P element activation. However, if this P sperm should fertilize this P egg right here, well, the P egg here has the anti-P element factors in here. So the P elements aren't going to become activated, and these flies, these zygotes resulting from this fertilization here, are going to develop normally. So what is the anti-P element factor? Well, those are going to be these things called piRNAs, so piRNAs. And as promised, in a future lecture, we are going to see why piRNAs accumulate in the cytoplasm here of the P eggs, and why they are sufficient to suppress P element activation in these flies right here. Oh, in the flies resulting from the zygotes after fertilization of the P egg. Okay, I hope that wasn't too confusing. Again, email me if you have any questions. In the next video, I think we are going to take a look at line and sign elements in the human genome. Then we move on to adenoviruses and gene therapy, followed by lentiviruses and gene therapy. And after that, I think we are going to take a look at coronavirus replication. At least that's my goal. Okay, well, see you in the next video.